Welcome back to the deep dive. I was going through the research for today, and I honestly had this moment where I thought, is this even allowed?
Huh. I get that. It does sound a little transgressive when you first hear about it.
Because we're not just, observing biology anymore, we're engineering it. We are talking about growing actual human hearts in a dish.
Well, not Frankenstein's monster in a basement. This is high-level precision stuff. We're talking about cardiac
Right,
organoids.
organoids. And before we even touch the how, which is just, it's wild, we have to talk about the why.
Yeah.
Because it seems like the way we currently develop heart medicine is, to put it bluntly, it's broken.
That's putting it mildly. I mean, look, cardiovascular disease is the number 1 killer globally. But the real problem is in the pharmacy. A new heart drug can take, what, 12 to 15 years? And cost over $1 billion.
Billion, okay?
And here's the stat that should really scare you. About 90% of those drugs fail in clinical trials.
90%. That's an atrocious success rate. If I did my job right 10% of the time, I wouldn't have a job.
Exactly. And the tragedy is why they fail. So often, it's cardiotoxicity. A drug looks perfectly fine in the lab, it looks fine in animal models, but then you give it to people and it damages the heart.
You're finding the problem when it's already too late.
Way too late.
So that's the big question. If we're testing these things for years, in petri dishes, on mice, how are we missing that a drug might stop a human heart?
It's because the old models are they're flawed. For decades, we relied on 2 main things. First, 2D cell cultures. So just imagine taking heart cells and spreading them out flat on a plastic dish.
Like a smear on a slide.
Exactly. But a heart isn't flat. It's a 3-dimensional pump. It has complex layers. It has pressure gradients. A 2D sheet of cells just doesn't act like a real heart. It's like trying to understand how a car works by looking at a pile of metal on the floor.
Okay, that makes sense. You lose the entire architecture.
Mhm.
But what about the mice? I mean, we hear about mouse trials all the time. We share a lot of DNA, right?
We share genetics, yeah, but physiologically, we're completely different engines. For one, a mouse heart beats about 9 times faster than a human heart.
9 times. So it's constantly revved up.
Constantly. And because it's running so fast, the whole electrical system is different. They have a totally different setup of ion channels. Those are the little gates that control the electrical pulse for each beat.
So a drug that messes with our rhythm, a mouse might just shrug it off.
Precisely. In fact, mice can tolerate about 37% of drugs that are actually toxic to us. That is a gigantic blind spot.
Okay, I get it now. The old tools are broken. So, enter the solution. The human cardiac organoid, or HCO. These things, from the pictures, they just look like little floating blobs, but they actually beat.
They beat. They can form chambers. They respond to drugs like a human heart would. It's a 3D model made from human cells. It's the closest thing we have to a tiny human heart avatar.
All right, so walk me through the recipe. If I wanted to build one, hypothetically, of course, where do I even start? Do I need a donor heart?
No, and that's the real beauty of this. You start with something called iPSCs. That's human-induced pluripotent stem cells.
I always stumble on that one. Break that down for me.
Think of it as biological time travel. We can take a regular adult cell from you, say a skin cell or a blood cell, and then we use this specific chemical cocktail to rewind the clock on that cell. We turn it back into a stem cell.
So it basically forgets it was ever a skin cell and becomes a blank slate again.
That's it. Pluripotent just means it has the potential to become any cell in the body. So the big trick is then convincing that blank slate that its destiny is to become a heart cell.
And how do you convince it? You can't just talk to it.
You use chemical signals, basically. There are a couple of ways to go. One is directed assembly, where you kind of force the cells into a mold, maybe a hydrogel, to give them a shape. But the really cool method, I think, is self-organization.
It sounds like magic.
Yep,
They just build themselves.
If you give them the right cues, they do. Cells have this incredible internal programming. If you flip the right switches, they'll spontaneously clump together and start forming the structures of an early heart. It's amazing to watch.
Okay, so what are the switches? The notes mentioned the GeneInCell protocol. It sounds more like a tech startup than biology.
It does, doesn't it? But yeah, it's pretty much the standard instruction manual. GeneInCell just stands for the small molecules you use to play with the Wnt signaling pathway. It's a 2-step process. Think of it as gas and brakes.
Gas and brakes. I like that. Walk me through it.
Okay, so step 1. You hit the gas, you activate the Wnt pathway. That tells the stem cells, okay, stop being generic, it's time to become mesoderm. And mesoderm is that middle layer in an embryo where all the muscle and blood comes from.
Right, so step 1 is become muscle potential stuff.
Yep. Then, just a few days later, step 2. You slam on the brakes, you inhibit that same Wnt pathway, and that shuts down all the other options and basically forces those cells down the cardiac path. Wnt on, then Wnt off.
What happens if you mess up the timing?
You get a mess, a chaotic blob of confused cells. It has to be very, very precise. You're also adding other things, other growth factors like BMP and activin A. These are called morphogens, and they sort of act like GPS signals, telling the cells where they are and what they should be building.
I saw a note about a breakthrough of that BMP signal. It led to something called cardioids.
Yes, that was a huge leap forward. By fine-tuning that balance between Wnt and BMP, researchers got the organoids to not just be a solid ball of beating cells, but to actually self-organize into a hollow cavity.
A chamber.
A chamber. It mimics the architecture of the early left ventricle.
That is incredible. So we're not just making heart muscle, we're making heart architecture. But a real heart isn't just one type of cell, is it?
That's a vital point. If you only grow the muscle cells, the cardiomyocytes, you have a twitching muscle, not a functional tissue. You need a cellular cocktail. You need endothelial cells, which line blood vessels. You need cardiac fibroblasts to build the structural scaffolding.
Glue that holds it all together.
Exactly. You need the whole team. Without the fibroblasts, it's too weak. Without the endothelial cells, there's no potential for plumbing down the line.
Okay, so we've mixed our cellular cocktail, we've hit the gas and brakes, we have a tiny beating blob, but there's a catch, right? The research says these organoids, they act more like baby hearts.
Even younger, actually. They act like fetal hearts, and this is the maturation problem. It's probably the single biggest challenge in the field right now. They tend to stay small, weak, and electrically immature.
Why is that? Are they just lazy because they're in a dish?
Part of it is actually their diet. Think about a fetus. What's its main fuel source?
Sugar, right? Glucose from the mother.
Exactly. So fetal hearts run on sugar, on glycolysis. But an adult heart, that thing has to pump nonstop for 80 years. It needs high-octane fuel. Adult hearts run on fat, on fatty acid oxidation.
So our little organoids are just stuck on a sugar high.
Pretty much. So the fix is to develop what we call maturation media. You take away the glucose and you feed them fatty acids instead. It forces them to make that metabolic switch. You're essentially starving them into growing up.
Tough love for cells.
And then you have to send them to the gym.
This is the part that I found amazing. We're building tiny gyms for cell blobs.
It's absolutely essential. A muscle needs resistance to get stronger. So researchers do things like grow the organoids around tiny, flexible pillars. As the tissue beats, it has to pull against the pillars. It's literal resistance training.
We're doing reps.
They are. And we also use electrical pacing. We use electrodes to zap the tissue, forcing it to beat at a faster, more regular rate. This helps mature their whole electrical system, especially their calcium handling.
Calcium. I know we need it for bones, but what does it do in the heart?
Calcium is the spark plug. The flow of calcium in and out of the cell is what physically triggers the muscle contraction. In immature cells, that flow is kind of slow and messy. Pacing trains it to be fast and efficient, like in an adult heart.
So we've got them on a keto diet, they're lifting weights, and we're shocking them. How do we even know if it's working? You can't put a stethoscope on something the size of a sesame seed.
No, you use some pretty high-tech surveillance. One method is calcium imaging. We use these fluorescent dyes that light up whenever calcium moves. So under a microscope, you can literally watch the wave of electricity spreading across the tissue.
Like a light show with every heartbeat.
It is, yeah. And then there's something called OCT, optical coherence tomography. It's like an ultrasound for tiny things, but it uses light. It lets you see the 3D structure and watch it beat in real time without having to cut it open and kill it.
Okay, so we can build them, we can mature them. Let's get to the so what? Why is this such a big deal? You mentioned drug safety.
That's the most immediate application. There's this huge global initiative called CIPA, the Comprehensive In Vitro Proarrhythmia Assay.
That's a mouthful.
It is, but the goal is simple. Stop approving drugs that secretly damage the heart. They want to use these human models to specifically screen for arrhythmias.
Irregular heartbeats.
And one thing in particular they look for is called QT prolongation.
Okay, what is that?
The QT interval is basically the time it takes for your heart's electrical system to recharge after a beat. If a drug makes that recharging time too long, that's prolongation, the next beat can come too early and the whole system descends into chaos. Your heart just flutters instead of pumping.
And that can be fatal.
Absolutely. And the scary thing is, Lots of drugs that aren't even for the heart can cause this. Antihistamines, antibiotics. They can accidentally block a specific ion channel called the HERG channel.
The HERG channel.
It's the master switch for that recharging process. And guess what? Mouse hearts don't rely on the HERG channel the same way we do. So a mouse test might give a drug a clean bill of health.
Well, it's a time bomb for humans.
Exactly. But the organoids, they have human HERG channels. They catch it.
And has this been proven? Have they caught something?
Yeah. Oh, yeah. They tested doxorubicin on organoids. It's a really common chemo drug, but it's notorious for causing heart failure down the road. The organoids replicated the exact toxicity doctors see in patients. The cells died, the beating got weaker. It proved the model was reflecting reality.
That's huge. And it leads to another idea, right? Disease modeling.
This is where it gets really personal. We can take cells from a patient with a specific genetic heart defect like hypoplastic left heart syndrome and grow a mini heart that has their disease.
So you can test treatments on a model of their heart without ever touching the patient.
Yes. It's also being used to screen for teratogens chemicals that cause birth defects. They tested the anti-nausea drug on dansitron.
For morning sickness.
Right. And the organoid studies showed that if it's used during certain windows of development, it can cause structural defects in the heart chambers.
Wow. That's crucial information to have.
And we saw it with COVID-19. Remember, early on, we didn't know if the virus was directly attacking the heart.
Right. Or if the heart damage was just from the stress of being so sick.
Well, organoid studies settled it. They infected the organoids with SARS-CoV-2 and proved, yes, the virus can directly infect heart cells and cause this massive inflammation, cytokine storm, right there in the tissue.
That's incredible. Okay, so what's next? What's on the cutting edge? I keep seeing this term heart on a chip.
It's the next evolution. An organoid is the tissue. The chip is a microfluidic device. Think of a glass slide with tiny channels like microscopic plumbing etched into it. You put the organoid inside.
And you can flow things through the channels.
Exactly. You can mimic blood flow. You can deliver drugs with incredible precision. There's one device they call the heart dyno, which is basically a 96-well plate where each well has a mini heart tissue pulling on flexible posts, so you can measure its contraction force in real time.
So it's mass production for drug screening.
That's the goal. But the really mind-bending stuff is when they connect different organs on chips. They call them multi-organ assembloids.
Wait, like a heart connected to a liver?
Yes, because your heart doesn't work in a vacuum. A drug might be totally safe for your heart, but then it goes to your liver, which metabolizes it and turns it into something that is toxic to the heart.
And if you only test on heart cells, you'd never know.
You'd miss it completely. By linking a liver organoid to a heart organoid, you can see that whole chain reaction play out. It's a body on a chip.
That's astounding. But I have to ask the obvious question. If we're making tiny beating heart chambers, why are people still dying on transplant lists? Why can't we just grow a new heart?
That is the ultimate goal, the holy grail. But we're hitting a huge physical barrier, vascularization.
The blood vessels again.
Yep. In your body, every single cell is just a hair's width away from a capillary getting oxygen. But in a dish, the organoid relies on diffusion nutrients just soaking in from the outside.
So the outside cells are fine, but the middle?
The center starts to die. You get a necrotic core. And that fundamentally limits how big they can get. We can make something the size of a pea, maybe. But a fist-sized pump, not without a functional plumbing system.
Is anyone close to solving that?
We're making progress. People are co-culturing vascular cells with the heart cells, trying to get them to form their own primitive vessel networks inside. These vascularized cardiac organoids survive better and grow a bit bigger, but connecting that tiny network to a major artery, that's a whole other engineering challenge.
So a full transplant is still a ways off. What about repairs?
That's much, much closer. That's the field of regenerative medicine. Instead of replacing the whole heart, think of it as a patch. After a heart attack, part of the muscle dies and becomes scar tissue, which can't pump.
And that permanently weakens the heart.
Right. So the goal is to grow a sheet of this organoid tissue and literally graft it onto the damaged area.
A living Band-Aid.
A living tire patch, yeah. And they're already testing this in models. They'll use cryo-injury, basically giving the organoid a mini heart attack by freezing one spot to study how it heals and if these patches can integrate and start beating in sync.
It's just amazing to think about the journey.
Yeah.
From simple twitching cells to engineering spare parts.
It really is. We're truly entering the era of precision medicine.
So bring this all home for us. What does this mean practically for someone listening?
It means the end of one-size-fits-all medicine. Your genetics are unique. In the future, if you have a heart condition, your doctor won't just try a drug and see what happens.
They'll grow my mini heart.
They could. They could take your blood or skin, make your personal cardiac organoids, and test 5 or 10 different drugs on them in the lab. They can find the one that works best for your heart with the fewest side effects before you ever swallow a single pill.
It takes all the guesswork out of it.
It does. And I want to leave you with one last thought. We started by talking about that 90% failure rate in clinical trials and the risk that human volunteers take.
Right.
Imagine a future where no human is ever the first to take an experimental drug. Imagine a clinical trial in a dish. Instead of 100 people, we use thousands of these little avatars, mini hearts, grown from the stem cells of people with diverse genetic backgrounds. We could find out if a drug is dangerous for a specific population before it ever harms a single person.
That's an incredibly powerful idea. The avatars take the risk for us.
It's not science fiction. That's where this is headed.
Well, my mind is officially blown. From rewinding cell clocks to heart gyms and body on a chip, the science here is definitely beating strong. Thanks for walking us through all this.
My pleasure.
And thank you for listening to The Deep Dive. We'll see you next time.